All right, so we've talked about DNA. We've talked about the fact that DNA is in every cell. And so what I want to talk about today is the fact that proteins are built from the information in DNA. This is what causes you to look the way you are. RNA is necessary for that. We call this idea, this connection between DNA, RNA, and protein, the central dogma of biology, the big idea of biology. So DNA is your genes, RNA is what it's copied into, and that causes proteins to be built, which causes your traits. And so DNA is transcribed into RNA, which is translated into protein. Structurally, a gene is a section of DNA. Functionally, a gene is the code for a protein. So we've talked about the fact that in biology, form follows function. The form of a gene, a piece of DNA, follows the function, which is the code for a protein. It's on the sense strand of the double helix, and when you looked at replication, you saw the orientation of a DNA 5' to 3' based on the location of the phosphate in the nucleotide. That becomes very important in transcription as well. The 3' to 5' strand is called the antisense or the template strand. This is the strand that's actually used for transcription. Template means the original that you modify or you copy, or the thing you're working from. Workbench strand. So the phosphate is attached to the 5' carbon in the sugar. Organic chemistry beyond the scope of what we're talking about, but they are numbered this way. The 3' is where the next nucleotide attaches to the phosphate of that nucleotide. Transcription literally means copying or writing somewhere else. The DNA unzips just like before, but rather than both sides being templates, only the template strand gets nucleotides, and these are RNA nucleotides. This finished RNA strand is called the transcript. It carries the DNA message out to the ribosome, and the message is instructions for a protein. So let's talk about RNA real quick. It has three main structural differences from DNA. The sugar in DNA is deoxyribose, but in RNA it's ribose. That's where the letter R and D come from. They're very similar, except there's a missing oxygen down there in the bottom right corner of the deoxyribose that is there in ribose. It's single-stranded instead of double-stranded. DNA is double-stranded because its mirror image allows for repair if something goes missing. RNA is a very temporary thing, so if it gets damaged, we don't need to fix it. We just trash it and start over. Also, there's no T in RNA, so A pairs with T in DNA, A pairs with U if we're talking about RNA. So uracil replaces the thymine. Uracil is an imposter T. RNA has many functions, whereas DNA just has one: store the code. For example, messenger RNA, mRNA, is the transcript. Ribosomal RNA makes the ribosomes. So the factories themselves where the messenger goes are made of RNA. Transfer RNAs bring in the amino acids, kind of like supply trucks or tow trucks. They drag the amino acids to the ribosome. And also there's a special class called ribozymes, which are enzymes, but they often catalyze reactions of RNA. Translation then is changing form. It's not just copying. We need a dictionary to do translation in languages, and you'll need a dictionary of sorts to do translation in the central dogma as well. So this is where we actually build the protein polymer, the chain of amino acids, from the nucleotide sequence in the RNA. Ribosomes are responsible for this. So we have these amino acids, and each code word or codon in the message calls for one amino acid. The code words are 3 letters long. So there are 64 combinations of 3 letters. And a molecule of transfer RNA matches up with the codon in the messenger RNA. That 3 letters in the transfer is called the anticodon. There are 3 codons which are literally stops and break the ribosome apart, so protein synthesis stops. Proteins do various jobs within the cell, but whatever the protein is supposed to do and be is dictated by the code in the gene, and it's carried to the ribosome to be built into that functional product, the protein, by the messenger RNA. So there's a picture showing that the DNA is in the nucleus, transcription happens in the nucleus, the messenger RNA leaves and goes out to the cytoplasm where we find ribosomes, and at the ribosome we build proteins. Keystone statement there. Sometimes we need additional processing. Sometimes mistakes happen. Cells are very reliable in DNA replication, but mutations can occur. The word mutation just means change. Some mutagens can't be avoided. A mutagen would be change causer, gen, to make or create. And so gamma rays, which is from the solar system around us, ambient radiation, rocks are radioactive. Sunlight causes mutations. You can't avoid these things. And so here's an example of some DNA damage which led to cancer because of sunlight damage in skin. We can prevent some causes of mutation. Too much sunlight, like the lady on the beach has, or x-rays. Sometimes you need an x-ray, like if you have a knife in your head, but if you don't get extra x-rays. You can always say no. Alcohol causes mutations of the DNA in the liver. Other carcinogens, such as those in tobacco. And those in tobacco are some of the most preventable, yet some of the most widespread carcinogens that cause DNA mutations, which loss of control of mitosis is what cancer is, and many of the genes that get mutations from these things control mitosis, and so you can develop cancer and need a surgery like this one, where this person is having their mandible removed. This is a preventable event because of the carcinogens in tobacco. So that's an avoidable cause of DNA mutation. Another keystone statement there. There are various kinds of mutations. Some of them happen at a single point in the DNA. We call those point mutations. Point mutations have some effects. There can be other things like duplication, inversion, deletion, insertion, translocation. Not all mutations cause the same effect. Some are silent, meaning that the DNA changes, the mRNA changes, but the protein doesn't, because of the redundancy in the codon code for amino acids. Sometimes multiple codons code for the same product, and you can get a change, but not a change in the protein. That's called silent. If we change where the amino acid is placed, or a different amino acid, we could maybe make something that does a similar job, but doesn't happen exactly the same. So a missense mutation still makes a protein, but it doesn't do the job quite the same. And a nonsense mutation doesn't make the protein at all. Frameshift mutations are almost always nonsense mutations. They cause where the words are located to change. And so here are a couple more examples. You can take a look at the slides if you want to see these in any more detail. And that's the end of that for today. Have a good week.